Good day, this is the Professor, CBMD PhD, and today's Monday, July 24th, 2023. It is 3:59 and 17 seconds, AM Eastern Standard Time, and today's research, polyfluoroalkyl substances. So let's come to it here. As polyfluoroalkyl substances reduce the activity of immune cells, study demonstrates immune modifying effect using a new method. Date July 18, 2023. Source: the Most Center for Environmental Research. Because they are persistent, current polyfluoroalkyl substances are also known as chemicals for eternity. They can lead to liver damage, thyroid disorders, obesity, hormonal disorders, and cancer. A team of researchers has now shown that polyfluoroalkyl substances can also reduce the activity of human immune cells and thus impair health. The method can also be used to reveal the immunomodulatory effects of other chemicals. Whether in cosmetics, coated pans, or outdoor clothing, polyfluoroalkyl substances are used in many everyday products. This is because polyfluoroalkyl substances are water and fat repellent, heat resistant, and extremely durable. As polyfluoroalkyl substances have been produced since the 1950s, and there are now around 10,000 different compounds, polyfluoroalkyl substances are poorly to hardly biodegradable, and that is a real problem. They therefore accumulate in the environment, in the soils and bodies of water that can even be found in Antarctica, that can enter the human body via food, drinking water, or the air. As studies have shown that polyfluoroalkyl substances can be detected in the blood of almost everyone in the world. What this means for our long-term health is not yet known. However, exposure to polyfluoroalkyl substances is known to cause liver damage, hormonal disorders, and lower birth weights. Many epidemiological studies have shown an association between increased polyfluoroalkyl substances exposure and reduced immune responses after childhood vaccinations. Could polyfluoroalkyl substances be affecting our immune systems? And if so, how exactly? What happens in the immune cells? And to find out exactly what happens at the cellular level after polyfluoroalkyl substances exposure, the researchers used a special immunological measurement method they developed. Using multi-parameter special flow cytometry, we can detect up to 30 markers in the blood sample using different fluorescent dyes and thus identify many different immune cell types and their activation. The team used immune cells from the blood of healthy donors. First, the isolated immune cells were exposed to different polyfluoroalkyl substances mixtures for 20 hours. In the laboratory, they selected 6 polyfluoroalkyl substances that are particularly common in the environment and prepared 3 mixtures. One mixture with 3 short-chain polyfluoroalkyl substances, one with 3 long-chain polyfluoroalkyl substances, and one with all 6 polyfluoroalkyl substances. The immune cells were then stimulated using standard activation methods. The researchers then determined how active they were at the cellular level by using multi-parameter special flow cytometry. The results of immune cells previously exposed to polyfluoroalkyl substances showed significantly lower activity than untreated cells. This was particularly true of T cells. For example, the T cells produce less of the messenger substances they normally use to communicate with each other and to recruit other immune cells or to trigger inflammation. The strongest effects occurred when all 6 polyfluoroalkyl substances were mixed. Here the effects of the different polyfluoroalkyl substances are clearly compounded. Polyfluoroalkyl substances reduce the activity of 2 out of 5 types of immune cells, in particular mucosal-associated invariant T cells and T helper cells. Mucosal-associated invariant T cells are formed in the mucous membranes and form the first effective defense response. If the activity of the mucosal-associated invariant T cells are restricted, it is much easier for pathogens to invade the body. As T helper cells are involved in the production of antibodies, if they are inhibited by polyfluoroalkyl substances, it is likely that fewer antibodies are produced, which could explain the reduced immune response to vaccination. As further investigations carried out at the genetic level were consistent with the results at the cellular level, genes that normally play a role in T-cell activation were downregulated after polyfluoroalkyl substances exposure. The study clearly shows that polyfluoroalkyl substances reduce the activity of immune cells if a person is exposed to high levels of polyfluoroalkyl substances. This is likely to be reflected in their health. For example, through a higher susceptibility to infections. Unfortunately, even if polyfluoroalkyl substances are completely banned, they will not disappear from the environment anytime soon. It is therefore important to gain a better understanding of the effects these substances can have on our health. In future studies, the team plans to simulate in vitro polyfluoroalkyl substances, mixtures, and concentrations as they occur naturally in human blood and determine their effects on immune cells. And so far, tests for immunotoxic or immunomodulatory effects are not part of the regulatory testing and assessment procedures for chemicals. However, as many diseases, from allergies to cancer, can be traced back to a dysregulation immune system, they believe this is urgently needed. They hope that with their study and their new and practical testing method, they can help pave the way for this to happen. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.